So, introductory neuroscience and neuroinstrumentation, different event-related potentials, part 2. So, now we will consider slow waves in the brain, specifically the CNV, the contingent negative variation, and the Bereitschaft potential, also called the readiness potential. Okay. Slow waves basically mean DC shifts. So one point to note is if you're going to record slow waves, you have to change your filter settings so that it's DC to 100 hertz or 30 hertz or whatever. If you put it at 1 or 2 hertz, you will to 30 hertz, which we normally do for other evoked potentials, you will miss the slow wave. Okay, so bear in mind that you have to record from DC to 30 hertz or 100 or 50 hertz because the DC will allow you to record slow waves. Okay. So preparing a moment or waiting for a stimulus that will show up in a few seconds is accompanied by a slow wave in EEG. You also get slow waves in epilepsy, and these are slightly different. This is pathological slow waves. We are talking here of normal slow waves. Okay? So there are 3 types of anticipatory slow waves that occur based on the experimental context. One is the Bereitschaft potential or the readiness potential. The other one is the contingent negative variation, and there is a stimulus preceding negativity. We will focus on the BP and the CNV. So the Bereitschaft potential and the CNV are due to the summation of a large number of postsynaptic potentials, EPSPs, in the cell columns of the cortical brain areas that will be involved in the processing of future stimuli or events. Okay. So consider the schematic on the right. It's over the premotor area, that is in front of the central sulcus you have the motor area, and in front of it you have the premotor area. So the Bereitschaft potential experiment is very simple. You tell the subject, please press the key of the mouse or a joystick or whatever whenever you feel like. Okay, there is no stimulus. The stimulus is from inside, from the person. So it's called a self-willed key press. So just before the person actually presses the key, okay, you get this slow rise in negativity, negative up. Okay, and the initial part is called BP1, Bereitschaft potential 1 or readiness potential 1, and it starts more than half a second before you press the key. Okay, you're only aware of the intention of pressing the key much later. Okay, and then there's a sharp rise, and that's BP2, readiness potential 2, and after that you have your key press, and then it goes down. So this whole complex is the Bereitschaft potential, this BP1 and BP2. So this is fascinating because before you're aware of the intent to press, the brain already has started doing it. So the question is, do you have free will? Because this was a huge philosophical question. If you're not aware of something, when the brain's activity can be recorded, that means the brain starts doing its stuff before you intend to do something. I mean, that's one possibility. So this was a very big controversy or a debating point, and it still is. One of the problems with the Bereitschaft potential is a very delicate potential. It rises over here, and you need a DC, recording amplifiers, and it's very, very sensitive to any kind of artifacts, AC noise and stuff. So this is one of the first experiments done in Austria, Germany, and you see the subject is inside a Faraday cage. So the Faraday cage is basically it's got a wooden frame, and it's chicken mesh, copper mesh inside, which is grounded. So it prevents interference, any electrical interference from outside. So it has to be, it can be recorded only with stringent recording conditions. It's not as easy to record as your auditory evoked potential or visual evoked potential. And part of the difficulty in recording contributed to the controversies because some people couldn't find it, so on and so forth. So again, the Bereitschaft potential is a premotor potential occurring prior to a voluntary movement, and it reflects preparation for upcoming voluntary movement. So it's a slow negative shift, and there are two components. There's an early BP and then there's a late BP. Okay. So the free will question, do you have free will or is there a green man inside your brain orchestrating things, has now come to rest in this because one of the things is our consciousness, the resolution of our consciousness is in hundreds of milliseconds. If you look at the P300 or even if you look at the MMN, it's the minimum time is 150 milliseconds. With the P300 it's about 300. So our consciousness is not, doesn't have a resolution shorter than that. We cannot resolve things less than 100 milliseconds or 10 milliseconds. So it may be a straw man, this whole free will question. It may be that a resolution of our, when we are of consciousnness is in chunks of 300 milliseconds. Okay. So it's not such a big deal that this, it starts at half a second before the key press. So this is a classic Bereitschaft potential. You have individual responses, very good recordings. There's no noise at all, and it's maximal over the opposite side of the scalp for upper limb movements, and comes in the midline for lower limb movements. That's because the way the hand and the leg is represented on the opposite side of the brain. So the following areas are activated during BP. One is the primary motor cortex, then the primary somatosensory cortex. This is on either side of the central sulcus, which you studied in macroscopic anatomy, and also the premotor cortex and 4 motor areas in the mesial frontal cortex, which is in between, the hemispheres, and it comes in between, so those areas in between. So let's consider the contingent negative variation. So this is very similar to the Bereitschaft potential. In the Bereitschaft potential, it's a self-willed key press. You press the key whenever you feel like. The contingent negative variation, on the other hand, is you press it, it's contingent to a stimulus before. So typically the experiment is like this. You have one stimulus called S1, which could be a click or a tone or a flash of light. And after about 2 or 3 seconds, which this is to prepare you, after about 2 or 3 seconds, you have the second stimulus, to which it could be anything, again a click or a tone or a flash, to which you make a key press. And between S1 and S2, you have this negativity. Here it's negative down, so it's kind of going down over here, but this is the contingent negative variation. And this was one of the earliest studies which showed a consistent pattern of electrical response related to a cognitive process, in this case expectancy. It was also the first ever event-related potential which was reported. Gray Walter is an American neurophysiologist who worked in England, and he was the person who found this potential. It is most prominent in the vertex, and it's symmetrical on both sides. And again, it can be divided into 2 parts, an early contingent negative variation and a late CNV. So the early CNV has a heat map distribution like so, and the late contingent negative variation has a heat map distribution of like so. So the generators, as I mentioned, are in the premotor cortex, Brodmann's area 6, and also some of these areas which are activated during the Bereitschaft potential are also supposed to be involved in the contingent negative in the generation of the CNV. Okay. So the thing about the CNV is it could be a possible biomarker for Parkinson's disease. Now Parkinson's disease, it's basically, there is a symptom called start hesitation. You tell a person to start walking, they take some time. Once they're walking, you tell them to stop, they take some time. They can't stop immediately, they have a starting problem. So these are classic symptoms of Parkinson's disease. It's a disease of affecting the motor system. And here you have a stimulus, S1, and you have to get ready, and then you have to press the button at S2, and there would be a delay in Parkinson's patients, or it would be abnormal, the CNV, I think. So it could be used as a biomarker for Parkinson's disease. Okay. So coming to another potential called error-related negativity. One thing to bear in mind with the contingent negative variation, the BP, and all the cognitive potentials is that they're all uniformly across the board depressed in disorders of higher mental function, for example schizophrenia or bipolar disorders. So these are very sensitive indicators of dysfunction, but they're not specific. So you have to bear that in mind. So coming to error-related negativity, consider a task. You have to make a correct response, to a task. It could be anything. Count the number of dots on the screen, for example. Now, if you make a mistake, you get a response called the ERN, the error-related negativity, which comes about 150 milliseconds, okay, after the mistake. Okay. And it is a component which is supposed to arise from the anterior cingulate cortex. This is looking at the hemisphere, the right hemisphere with the left hemisphere removed, looking at it from the left side. And this is the area, the anterior cingulate cortex. Anterior cingulate cortex is a much storied cortical area. Some neuroscientists feel that one of the seats of consciousness, as it were, is in the anterior cingulate. Okay. So this ERN is a ERP component reflecting the response monitor system, and it tells you whether you made a mistake or not. So you can imagine it's useful to know that you made a mistake. Okay. A very similar potential related to ERN is the FRN, which is feedback related negativity. This occurs when an individual receives external feedback. It could be either visual or auditory, indicating that the performance is worse than expected. So for example, consider a simple gambling task. So you have to gamble, and you have two responses, and if you choose one, you will always get a reward. If you choose another, you may or may not get a reward. Okay, but when you do get a reward, it's huge. Like for example, the first time you choose the response, the low reward condition, you get 10 rupees. The second time, the second condition is, and you get it 95% of the time, 10 rupees, always. When you choose it, you get 10 rupees. But if you choose the other option, you get 1,000 rupees, but very rarely. You don't get it always. So this, what you get is, the index for this is the feedback related negativity, and it occurs around between 150 to 300 milliseconds. So on this graph on the right, you see a negative outcome which is shown by the dotted line, and the positive outcome which is shown by the solid line. So the negative outcome between 150 to 250 is the feedback related negativity. Okay, so it comes as indicated in paradigms which include monetary loss, feedback about performance in simple games, etc., and it's very similar to the earlier error related negativity. Okay. And you look at the heat map on top, it's fronto centro parietal distribution. Okay. So typically it occurs 250 milliseconds after feedback, and as I mentioned, it's the central frontal areas. And again, the generator most probably is the anterior cingulate cortex. It's difficult to figure this out because it's deep, so you have to do source modeling, which we'll talk about in our next lectures. So consider the data on the right. So you have FRN differences, loss win, in two kinds of patients. One patient is one set of patients were obsessive compulsive had obsessive compulsive disorders, and the other set were normal. So the obsessive compulsive disorder SOC group is in red, and the normal or the low obsessive compulsive is in blue. And you see the obsessive OCDs, they have a huge FRN. Their brain reacts majorly to a loss, and so again, these things, these kinds of potentials could possibly be used not only to screen obsessive compulsive disorders, but also to see if therapy, they get various drugs to see if therapy makes a difference. Okay. Coming to another potential, and now this potential is very interesting. It's present at birth. It's called P50 gating. So consider two clicks, click, click, okay, and there's a huge response to the first click, okay, but the response to the second click is kind of muted because there's a little bit of habituation or whatever which goes on. Okay, so the P50 event occurs approximately 50 milliseconds after presentation of a stimulus, usually an auditory click. So you have one P50, and then you look at the P50 to the next second thing, and so here on top, on the right, you see the response to the first sound, okay? And you see the response to the second sound, it's been gated or it's been filtered. Now, when you have poor gating, the first response isn't so big, but the second response is neither small. So typically, you see this in disorders of higher mental function like schizophrenia. And this has an evolutionary significance that you can't have the same kind of response to redundant stimuli, okay? So in schizophrenia, consider these data over here. You have the controls. This is the first click, the amplitude of the P50, the auditory response following the P50 gating. And then you have a second click, and it's smaller. But in schizophrenia, the first one is this big, and the second one is also this big. Okay, so it means that schizophrenics have difficulty in controlling the input of stimuli. So if I'm talking to you, one hopes you're paying attention to me, but there are a lot of stimuli in your environment, in your acoustic space. A schizophrenic will pay attention to everything. He or she can't gate, and so there's a barrage of sound always, or a sensory input coming to the schizophrenic. Okay. And the other interesting thing is healthy infants show this response even when they're between 1 and 4 months, even when they're very young. And this shows that sensory gating is present early in development. And the other places where P50 gating can be abnormal is in traumatic brain injury, recreational drug use, and post-traumatic stress disorder, like veterans, army veterans. They have these problems. They cannot filter, stimuli. Okay, and we considered early on in a previous lecture using linguistic stimuli as language disorder probes, where we did LPC monitoring, late positive complex P1, N1, P2 monitoring in tribal children in Chhattisgarh. Another way to use this is using the recognition potential, which was discovered by Professor Alan Rudell in SUNY Brooklyn, one of my mentors. And he showed that mother tongue alphabets can be recognized as early as 120 milliseconds, in the range of 150 to 300, but as early as 120 milliseconds in occipital electrodes behind. So this could be useful as a biomarker for dyslexia. So if you see over here, this is Alan, you see the RT. RT is reaction time, okay, where you have to respond if you guess correctly, and it's useful to decrease the signal to noise ratio in the data by using only the correct responses. And this is the recognition potential, and this is the negative up, and this is the recognition potential window. Okay, you only get it with your mother tongue or with a language which you're very familiar with. Suppose I flash Chinese vowels to an Indian child, there won't be any RP. Okay, so we did some pilot experiments in St. John's, where to a Hindi-speaking subject we showed Hindi vowels and Kannada vowels. I'm sorry, to a Kannada-speaking subject we showed Hindi vowels and Kannada vowels. So the black trace is the response to Hindi, and the thickness of the trace indicates the standard deviations, the response. Likewise for the Kannada, which is red. And if you subtract both of them, you get the RP, which is the thin blue line. So you can also use it in an Indian context with Indian alphabets, the vowels, the consonants, and the conjuncts, and you would get the RP. So again, this would be useful in an Indian context, because many Indian children learn English late, but they learn their mother tongue first, and we could use their mother tongue to see screen if they have dyslexia or not. Okay. And you might say, what do you do if you find a child has dyslexia? It's pretty easy to reverse. You give them computer games and with feedback, and you train them. And within about 3 weeks, it makes a difference. They learn, their responses are good, and they get the RPs for their mother tongue. Okay. So one more, N170. This is for face recognition. So typically on the right side, the right occiput, there is a face recognition area, and this is a face-sensitive event-related potential. It's characterized by a negative deflection at around about 170 milliseconds. This is it. And images of faces or even face parts such as eyes, they elicit an enhanced N170 in comparison to other visual stimuli like animal faces or body parts or cars or buses. So this is very specific to face and eyes, and it's maximum over the occipitotemporal area and more on the right side than the left side. So here on the right you see a typical N170 experiment. So the green trace are stimuli which are not face related like plants, etc. The red is face stimuli, and you can see a huge response, and this is the heat map on top showing that most of the activity is in the back and specifically on the right side. So much of this work was done by a very good French scientist, Stanislas Dehaene, and for those who are interested in more, I encourage you to go and check him out on PubMed. Okay, so thank you. That ends our lectures on event-related potentials.